Hi, everyone. Welcome to the Johns Hopkins seminar series on the cerebellum. Today, as our guest, we have Jean Lorenz. Jean was born in France and earned his PhD from College de France and was mentored by Jacques Droules. His research during his PhD focused on theoretical modeling of the vestibular system, demonstrating how Bayesian inference can help explain the link between sensory noise, statistics of head movements, and perception of self-motion. During his postdoctoral training with Bernard Hess and Dora Angelaki, he transitioned to electrophysiology of the vestibular system, vestibulo-cerebellar system in macaques and in mice. He now leads a research lab at Frankfurt in Germany, where he develops naturalistic wireless electrophysiology in mostly rats and marmosets. Sean, it's wonderful to have you.
Thank you, Reza, for the introduction. And before starting my talk, I'm going to just give you a glimpse at my laboratories. So this is what we are developing, free moving rat and marmoset. For the rat, we are already doing wireless recording in a pretty large arena. So that's 5 meters across, wireless neuronal recording. And for the marmoset, we continue setting things up with really a focus on freely moving naturalistic behavior. And with that, I go to today's topic, a tale of two vestibulo-cerebellum, and that's a presentation that I prepared with my student, Deepak, who's really a great and passionate student. So, if you read some papers about the vestibulo-cerebellum in the last year, you probably saw that they are talking about a lot of things like internal model and Kalman filter and active versus passive motion and gravito-inertial ambiguity and so forth. And my goal today is to put a little bit of order in all of this by offering a review/perspective about internal models in the vestibulo-cerebellum. And so that's going to be a systems neuroscience review/perspective with emphasis on theory, modeling, electrophysiology, and anatomy. So the vestibulo-cerebellum here in singular, but the title of my talk, A Tale of Two Vestibulo-Cerebellums, kind of implies that there are multiple of them. And indeed, this is the way I see things. There's not only two, but actually three vestibulo-cerebellums. And these are the nodulus uvula, the flocculus, so they are the same lobe, but definitely two completely separate regions. And then nowadays we see the anterior vermis, who is also called in the literature a vestibulo-cerebellum. So there are three distinct vestibulo-cerebellum, and today I'm not going to talk about the flocculus, which is really about eye movement. I'm going to focus on the way the anterior vermis and the nodulus uvula implement internal models of self-motion. So here's an overview of the plan of my talk. I will start by presenting the concept of internal model for vestibular information processing. And then I will talk about how these models are implemented in the vestibulo-cerebellum, and then I will have some time for conclusions. So let's start with some very basic things. What is the vestibular system? Well, it's something that originates from the vestibular organs in ear. They are in the inner ear, right next to the hearing organ, and they contain two types of motion sensor. The semicircular canal that sense rotation and the otolith that sense inertial motion. And there's a clear analogy. You can think of the vestibular organs like the little gyro chip of the brain. Now we have gyro chip everywhere in our smartphone and in drones and so forth. And if you know a little bit how they work, they don't actually tell you everything about the motion. What you have is that you need to read out the output and drive it into some software in order to extract the actual motion of your phone. Well, that also holds for the vestibular organ. They are not telling you everything about head motion, and the brain has some algorithm to process the information that comes from them. And now that we are talking about algorithm for self-motion sensation, I'm going to introduce the most famous of all this algorithm, and that's the Kalman filter model. So the Kalman filter was developed in 1960 by Kalman, and it became popular very fast because it was used by the NASA to control the Apollo rocket. And pretty rapidly in the 70s, two researchers at the MIT started to realize that the Kalman filter was actually a great way to understand how humans sense self-motion. It was also great when it was applied to understanding motion sickness. And well, long story short, it took, I would say, 3 generations and about 30 years, but eventually Kalman filter became the most influential and widely accepted model of self-motion processing. So Kalman filter internal model, what is the big deal about this? What's the actual definition of this? I think it's good here to make some points very clearly. So I'm going to do something I usually don't do. I'm going to read the text from my slide. So an internal model is a neural representation of how the body and sensory system behave that mirrors their behavior in the real world, right? So that's the important thing. It's an internalization of the physical rules that govern motor and sensory system. So the Kalman filter is based on an internal model. So this one type of model based on the concept of internal model, so to speak. And another important property is that it's a normative approach. And it means that you start from an objective description of the body and sensory system. In fact, you start from an objective description of the real world, and that's why it's objective. And then based on that, it tells you what the brain should do to estimate motion optimally, as accurately as possible. Now, why is it that there are so great models of the brain? I'm not trying to claim that the brain is literally doing matrix operation and Kalman filtering. I think the point is that in many cases, the brain actually senses motion very accurately, and by doing that, it approximates Kalman filter prediction. And this in turn means that Kalman filter help us interpret and predict what the brain does. And another thing which is great is that Kalman filter help us pinpoint key computational steps which are in the Kalman filter algorithm and that the brain necessarily also has to implement. And once we know them, we can look for neuronal substrate of this. So I wanted to make these points quite clearly. And now let's have a look at the Kalman filter algorithm in general. Well, the idea is that you have an internal model of hand motion, just as I explained. And this internal model can be updated in two ways. The first is by integrating motor reference copies. So there's the hypothesis that the brain knows how motor activity will alter hand motion. And then the other is a sensory pathway that works as follows. First, the brain is going to transform the internal estimate of motion into sensory prediction. So it's going to simulate how the sensor, what the sensor should feel. And then it's going to compare the sensory prediction with the actual sensory signal. And in the case of a sensory prediction error, of a mismatch, it's going to correct the internal estimate of motion through feedback signals. So that's the general outline. And let's go a little bit more into detail. How specifically do you make a Kalman filter of hand motion? Well, the first thing is that you have to define the relevant motion variable. And in the case of vestibular information processing, this is rotation velocity, tilt relative to gravity, and translation. And all of this tells you what you need to do for vestibular science of the head. However, another great thing about Kalman filter is that they are extremely modular, and you can simply add more motion variable, for instance, neck and body motion. So then let's talk about rotation, or actually let's not talk about this. I want to save time, so I'm not going to go into the detail, and I will just say you can make a Kalman filter of rotation that will integrate motor reference copy about head rotation and sensory input, mostly the semicircular canal and visual input, because you can see your own rotation. I don't go into the math, but I'm going to show a couple of classical experimental results. So the first is rotation in darkness. So the experiment is that you take a monkey, you put it on a rotating chair in complete darkness, and you rotate it at a constant velocity for a minute. And when you do that, you're going to activate the semicircular canal, and if you monitor the activity of the canals, it's going to decrease in about 15 seconds. So the canals are high-pass filter. If you monitor the perception of the monkey, you can do that by measuring the vestibulo-ocular reflex, then you're going to see that it's decreasing, but it is decreasing slower. And that difference in dynamic, this increase in the time constant, can be explained by a Kalman filter. Another interesting thing is that if you stop the monkey, then you will get an after effect. That's pretty obvious. Another classical experiment, now you rotate the monkey in light and you will see that rotation perception persists indefinitely. And if you switch off the light without stopping the rotation, you will see that it decreases kind of slowly like that. All of these are classical experimental results that can be explained by a Kalman filter. Now we're not going to focus so much on this because none of this is actually happening in the cerebellum. Let's now focus on tilt and translation. And the first thing here is to note that tilt is always going to be the consequence of rotation. So that is because whenever you are making a tilt motion, you are changing the tilt of the head, you are actually making a rotation. And you can express that by a simple equation. Tilt is the integral of rotation, and that is going to go into your internal model. As far as sensation is concerned, tilt and translation are both sensed by the otolith organ. The best way to explain this is a little analogy. The otolith organ are a bit like as if you had a pendulum attached to the head. And if you have a pendulum fixed to your head and you tilt from side to side, then in an egocentric perspective, the pendulum will appear to swing from side to side. And if you translate from side to side from an egocentric perspective, the pendulum will also swing. So we can summarize this by a single equation. The otolith sends the sum of tilt and translation, and here tilt really means the gravity vector in an egocentric point of view, which will change in an egocentric point of view whenever you tilt, and translation means translational acceleration. And so in our Kalman filter, we are going to have a prediction of this, an internal model of this, tilt plus translation. And then it's going to be compared to the otolith signal, and it's going to, whenever there's a mismatch, it's going to drive some feedback loop. And here there's already quite a few boxes and arrows, and I want to save a little bit of time. So I'm just going to say that if you boil it down to this, then it pretty much simplifies to these two equations. Tilt is the integral of rotation, and translation is equal to otolithic signal minus tilt. So the Kalman filter predicts exactly that, but it also predicts a little more. It predicts, so the Kalman filter can also integrate efferent copies. And then the Kalman filter is going to predict two other feedback pathways. So the first is a feedback pathway to tilt. And the idea is that if you create a long duration, low frequency otolith prediction error, which you can do, for instance, with a centrifuge, then you can create an illusion of tilt. And indeed, this is something well known, that you can create illusion of tilt with centrifuge or aircraft or things like that. The other is a little bit more complex, and it's a feedback to the rotation velocity. And now the idea is that if you have an incorrect rotation signal, which is creating, for instance, an incorrect tilt signal, then you might want to correct it. And there is one well-known example of this. Remember this experiment where you rotate the monkey and then you stop, and now the monkey has an illusion that it is rotating, even though it is not. The variant is that you rotate the monkey and then you stop and you tilt him, and now the monkey has an illusion that it is rotating around the tilted axis, which should cause him to tilt around. And when you do that, you actually create an otolith prediction error, and what you see is that the rotation after effect is shortened. Now I would like to point out that what you see here are simulations. These are actually real simulations that I made with a real Kalman filter, but these are also experimental results. You can really do that. It's been done a lot of time and it works. And it's bringing me to one point, which is that all of these little boxes and arrows in here, They are really equations, and they are actually the backbone of a series of quantitative models that emerged starting from Glassauer and Merfeld, later me and Karmali. All these models they are quantitative, they are accurate. You can put monkeys into 3D rotating chair, into centrifuge. You can do that with people, and you can predict exactly what they are going to feel or the vestibulo-ocular reflex. Even more, this model recently have proven extremely efficient at predicting the consequence of galvanic vestibular stimulation or vestibular lesions. So I talked about Kalman filter, rotation, tilt, translation. Another important aspect is the processing of active motion. Whenever you are moving yourself, you're going to have efferent copies about your motion. And as long as these efferent copies are accurate, then your estimate of motion will be accurate, and there is not going to be any prediction error. So note, however, that the fundamental operations that you will use are exactly the same that you use during externally generated motion. You still need this 3D integration in here, and you still need to run your forward model. However, this has implications for neuronal coding. You can understand that if some neurons encode sensory prediction error, then they are not going to respond the same during active and passive motion. And I think all of you know very well what I am alluding to, and we will come to that.
Bye.
Finally, last point, you can actually, so everything that I presented so far was in a head reference frame, but you can also add body reference frame to your Kalman filter. So basically you take it, let's make a little bit of space, and you can just add additional variable like the motion of the body in space, the motion of the neck, so head on body motion, so to speak. You're going to move the efferent copies in here because now all of the rotation will be the consequence of that. You're going to add additional sensory modalities, and there's going to be additional internal model, like of proprioception. There's going to be proprioceptive error. And you can make a nice big Kalman filter with all of this together. There's going to be quite a lot of equation, but it has been done. I did it. That's in my 2017 eLife paper. So that's about it for the concept of internal model. And to recapitulate, so what I talked about were internal model, how you sense rotation, how you process your otolith signal and sense tilt and translation, active versus passive motion, head versus body motion. And now I think the interesting questions are first, are all of these things actually relevant? Does the brain do all of these things? And then the next, if yes, then how do these things map onto cerebellar circuits? And I am going to spend the rest of the talk discussing about this, but just to answer that real quick, the answer is yes, 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 and yes. And let me just mention it that the internal model is now universally accepted as the best way to model vestibular information processing. Rotation sensation has been so much studied and modeled since the 60s that there isn't any debate about that. Somehow the notion of otolith internal model and tilt translation discrimination was controversial until the 90s, but now there's simply also overwhelming experimental proof that the brain does it. And I would also point out that it is indissociable from internal model. And what I mean by that is that if you accept there's an internal model of the vestibular organ in the brain, then there has to be an internal model of the otolith organ. And once you have it, then the brain's ability to discriminate tilt from translation is a fairly straightforward mathematical correlate. Regarding active versus passive motion, yes, there's extensive proof that the brain processes this thing differently from neuronal recording and also models of motion sickness. And finally, the matter of head versus body motion, it's fairly obvious that the brain can control the head and the body, and it's also been studied a lot. And with that, we're going to start discussing how all of this is encoded in the vestibulo-cerebellar pathways. And so let's start by recapitulating all of the circuits that surround the nodulus uvula and anterior vermis. So, okay, let's first put them right here in the middle of the slide. And then right now, I'm going to add the vestibular nuclei to that slide because they're quite important. So the vestibular nuclei are brainstem nuclei divided in 4 regions. Medial, that's the most important in here, as well as superior and inferior. And then another region, the lateral vestibular nuclei, actually laterodorsal to be accurate, which I put aside and we will see why in a moment. So the vestibular nuclei, especially the medial, are highly integrative, highly multisensory nuclei. So they receive vestibular organ projection, vision, proprioception. They do have a large variety of neurons. They drive eye movement, perception, and so forth. And so I try to convey all of this heterogeneity with the little rainbow color code in here. Okay, I'll put this aside for now. Next, so the vestibular organ project to the nodulus and uvula, but not to the anterior vermis. However, both nodulus uvula and anterior vermis receive projection from the medial vestibular nuclei. The anterior vermis also receives a lot of projection from the spinal cord, so these are the spino-cerebellar pathways. Yeah, so we have now the muscle fiber input. Then the nodulus and uvula project to the fastigial nucleus, and to be specific, to a little region in here, in the fastigial nucleus, named F4 in Fujita and all, which in turn will project to the vestibular nuclei. And the nodulus uvula also project directly to the vestibular nuclei. The anterior vermis project to the rostral fastigial nuclei, right here, and inter both this and that project to the lateral vestibular nuclei. In turn, the lateral vestibular nuclei project back to the spinal cord. So here we see a little spinal loop emerging, right? And still, the rostral fastigial nuclei also project to the medial vestibular nuclei. So we have the cerebellar cortex, we have the cerebellar nuclei, we have the muscle fiber input, and of course, let's not forget the most important, the inferior olive. So the nodulus and uvula is innervated by the beta nucleus in the inferior olive, and interestingly, all of the input of the beta nucleus appear to come from the nodulus and uvula itself, either through the beta fastigial nucleus or through the vestibular nuclei. And here there's even a small subregion of the vestibular nuclei named the parasolitary nucleus that seems to be a relay between the nodulus and uvula and the beta subnucleus of the IO. So in other words, we see a closed loop in here. For the anterior vermis, the projections are from the medium accessory olive as well as the dorsal, and this receive spinal cord projection, the spino-olivary cerebellar pathway, as well as input from the rostral fastigial nucleus. And to finish the diagram, the medial vestibular nuclei project to the spinal cord. And so that's going to be our basic circuit. So we see really 2 units well separated, like the nodulus uvula on one side, the anterior vermis, and the rest on the other. And now let's start talking about this region. We'll start with the nodulus and uvula, but in practice, I'll be switching between the two of them quite a bit. So most of the recent studies on the nodulus and uvula were made by Dora Angelaki group in the context of tilt translation discrimination. And as I mentioned earlier, like the whole model in that context simplifies to 2 simple operations. Tilt is sensed as the integral of egocentric rotation signal, and translation is sensed as the difference between otolith signal and that tilt signal. And this is what Dora has been studying since the early 2000s. I joined her in 2010. And the type of stimuli that we were using was fairly simple, like simple tilt and translation of the head that were always matched such that they activated the otolith in the same way. And that's an illustration of a classical translation selective cell. So what you do is that you translate your monkey from side to side, and you have here a firing histogram, and you see that the cell fires in phase with translation, obviously. This is the same cell recording during tilt, and here there is not much of a response. So Dora started rediscovering these cells in the early 2000s, and in 2010 I discovered tilt cells that will respond during tilt. So here we are tilting the monkey from side to side, and not much during translation. So at the population level, in the nodulus and uvula, I found that about 1/3 of the cell encode preferentially tilt. And so if you plot their tilt response versus translation, well, they will respond more to tilt, right? They are above the diagonal. About 1/3 encode translation, so they end up below the diagonal. And the rest are basically intermediate cells. That's a result that has been replicated quite a lot of times since. So here is a study from Trestay in mice. So these were macaques, obviously. And in mice, when we replicated the study, we saw exactly the same thing. Actually, if you look at the axis, the clusters are in exactly the same position. And recently, this has been replicated by Andrea Green Lab in Montreal in macaque. And once again, exactly the same result. So altogether, this is a really strong support for this internal model computation. However, here I want to emphasize something. At this point, we had not demonstrated one critical step, which is this three-dimensional integration. And what I mean by that and why is that important, I'm going to explain it. So during the tilt stimulus, we were tilting our monkey from side to side, and we found a cell that was responding. But for all we know, it could be simply a cell that encodes egocentric rotation velocity around the roll axis. Otherwise stated, a simple semicircular canal afferent. So there's really nothing so remarkable about having a cell that responds specifically during tilt. The important control was to demonstrate that the same cell actually encode allocentric tilt relative to gravity as opposed to egocentric rotation. And the way I did that was by designing another protocol named off-vertical axis rotation, where you tilt the monkey first, and then you rotate at a constant velocity around the head vertical axis. And when you do this, the head is going to alternate from right ear down to left ear down to right ear down to left ear down, exactly like during roll tilt, but by using a completely different egocentric velocity. And so here is the same cell. Whoops, I forgot to shut off the sound during tilt and during over. Well, I guess you can enjoy the sound of a neuron. And what you see is that it's responding whenever the monkey is right ear down. All right, and so that was the clear experimental demonstration that these cells in the nodulus uvula actually do a three-dimensional integration of rotation signal. So Dora and her group and her successor did quite a lot of experiment, and long story short, they identified translation codic cells in the nodulus uvula, in the medial vestibular nuclei, and also on the other side in the rostral fastigial nucleus. I identified tilt cells in the nodulus and uvula, and here one has to be a little bit careful. There has been report of tilt cells in the rostral FN and the vestibular nuclei. However, these studies actually didn't do this kind of 3D protocol, so we don't know for sure that this is allocentric tilt as opposed to egocentric velocity. Yeah, another thing, so there's one region where no one really recorded, and that's the beta nucleus of the inferior olive. However, as you well know, when you record in the cerebellar cortex, you can isolate simple complex spike, and since this complex spike comes from the inferior olive, when you isolate complex spike, it's basically as if you were recording inferior olive neuron. So a few years later, I looked at all of my recordings, and here is an example, the tilt cell as before. And you can see this nice big complex spike in here and in there, and notice that they are modulated in phase with acceleration. And there was a surprise. What I found is that the complex spike of all of the cells, that is tilt cells and translation cells, well, the complex spikes were always translation selective. A bit as if the inferior olive contains only translation cells and no tilt cells. Now that was a bit of a surprise. Why would tilt cell receive complex spike that encode translation And that got me confused for quite a while, but eventually I built the following interpretation. And the interpretation is the following. The nodulus uvula is trying to build a forward internal model of the otolith. It is trying to predict otolith signal. And now during tilt, there is one thing it can do. It can use egocentric rotation signal and integrate them in order to predict tilt. In other words, what the tilt cells are doing is that they learn to predict the gravity component acting on the otolith. On the other hand, during translation, well, only the otoliths are activated, so there is nothing that the brain can use to predict otolith signal. And therefore, what translation cells do in this situation is simply to encode the remainder to encode a sensory prediction error. One point in favor of this interpretation is that sensory prediction errors are associated with motion sickness, and as it turns out, translation are one way to induce motion sickness. Now, if you have a prediction error, what can you do about it? Well, maybe you were experiencing a translation and it was completely unpredictable and there was nothing you could do about it. But maybe it could be that your entire circuit was not tuned properly, and then you might want to just send that prediction error signal back to the entire circuit in order to drive adaptation. And based on that intuition, I built a neuronal network that actually starts completely randomly initialized and can learn to develop tilt cells based on its own output as an error signal. So this is coming towards an idea that the nodulus and uvula is indeed implementing a forward internal model of the otolith. It is outputting otolith prediction error, and it's actually using its own output to train itself. Now, some more argument in favor of this. Let's zoom back again to the standard model. And my hypothesis here is really that the nodulus and uvula is implementing the entire part of the forward internal model. And there are some additional experiments in favor of that. The first, is the somatographic feedback. We actually demonstrated that during low-frequency translation, the somatographic feedback can be observed in the activity of tilt cells. Second, this velocity feedback. Well, you remember that gravity dumping experiment where you create a canal otolith conflict that creates an otolith prediction error. Well, as it turns out, we did that with DORA, and we found that it activates translation cells, again confirming that translation cells might encode an otolith prediction error. And another argument, a classical one in favor of that, is that if you lesion the nodulus uvula, then you do abolish the velocity feedback. So to conclude about the nodulus and uvula, I wrote a perspective in 2022 where I named it the otolith vermis. And the reason for that is that everything we see in the nodulus uvula seems to revolve around inertial motion and otolith processing. And if we really go into the detail, we see that neuronal activity and the effect of lesion completely match the standard model of vestibular information processing, which I find quite amazing when we know that this model really originates in the 90s. In all events, we have a direct experimental demonstration of the transformation from egocentric velocity to allocentric tilt in 3D. And I would like to emphasize that all of this is based on externally applied motion, and this is going to be relevant a little later. And I think now let's move on to the anterior vermis. The first thing to say about it is that it is really, I think we can call it a spinal vermis, right? Most of the input to the anterior vermis are the spino-cerebellar tract, and most of the output are going back to the spinal cord. In fact, one can even wonder why these descending pathways are named vestibulo-spinal tract to begin with. The reason they are named like that is because they originate from the lateral vestibular nuclei. And why is the lateral vestibular nuclei named like that? Well, good question, because it does not even receive primary vestibular projection. It's really kind of an anatomical mistake of history. But, well, so in addition to that, you have a whole body of recordings of the anterior vermis in the context of locomotion in the cat, and what you will see in there are basically leg motion signal. So the anterior vermis is a spinal vermis, but this is not to say that it does not receive vestibular input or it does not do vestibular processing. It does, and that has been studied most prominently by Cathy Cullen's lab. And so we're going to look at some of her classical results. And the first one, let's look at a study made by Brooks and Collin showing evidence of vestibular motion sensing in the anterior vermis. So what they did is that they had the monkey on a chair and then they could rotate the entire monkey, the body and the head together, or they could rotate the head of the monkey only above the trunk of the monkey, or they could rotate the body of the monkey only, so they would fix the head and rotate the body under the head. And when they did that, they identified two types of neurons, some that were encoding head motion specifically, responding to whole body and head only rotation, or some that would respond to body and whole body and body only rotation, so body coding neuron. Note that the two types of neurons are activated during whole body vestibular only rotation, and therefore that's a direct evidence that yes, there is vestibular sensing of motion in the anterior vermis. They were named also unimodal and bimodal because this one, if you look carefully, you see that basically they only respond to vestibular signal, and the bimodal one, they respond to vestibular and proprioceptive signals. At the level of the entire network, they identified head and body coding cells in the rostral fastigial nucleus. That was basically Brooks and Collin, a whole series of papers. Recently, they reported head and body coding neurons in the anterior vermis. And is there anywhere else? If you look at the classical old paper from Cathy Cullen, you will find that in the vestibular nuclei, they identify only head coding neuron. So now this is all during rotation. What if you do the same with translation? Well, you can do it. However, here I think the studies were more looking at active versus passive, not so much at head versus body. But if you look carefully at the study, it seems that they reported only head coding neuron for translation. So this story of head versus body coding in the anterior vermis doesn't end here. There's also a very nice study coming from Andrea Green's lab. They have this wonderful system here with a 2D rotator on top of a 6 degree of freedom motion platform. And what they did were three-dimensional translation of the monkey with the head positioned at various static orientations. And what they found, I color-coded it a bit. So they analyzed their result basically by computing displacement index. Displacement index of 1 means that the cells are head coding, and the nodulus and uvula, it was mostly head coding. In the anterior vermis, they found a combination of in the rostral fastigial nucleus, they found a combination of head and body coding, and in the vestibular nuclei, only head coding. So to conclude with that, we see mostly head and body coding in the anterior vermis, and pretty much only head coding in the nodulus and uvula and in the vestibular nuclei. So the other fundamental result from Cathy's lab, of course, is the coding of externally generated motion. She made a series of studies which are really famous, where basically she demonstrated that most neurons are sensitive to externally applied motion, but not to self-generated motion. The interpretation being that this neuron actually encodes sensory prediction error, so this can be canal prediction error or otolith prediction error. I think I don't need to explain the study too much because that's really famous. And to recapitulate, in the context of rotation, they found neurons that encode externally generated motion specifically in the vestibular nuclei, anterior vermis, and rostral fastigial nucleus for rotation, and in the rostral fastigial nucleus and vestibular nuclei for translation. So to conclude with the anterior vermis, I think it's fair to call it a spinal vermis because it's really connected to the spinal cord. However, there are clear vestibular input with vestibular sensing of externally generated head and body motion. And it feels like this specific coding of externally generated motion is then carried over to the vestibular nuclei. And all of this matches internal model computation. This can be shown using the model I published in 2017. And I think to get this a little bit more in the context of motor control, there are some very beautiful modeling studies from Glassauer, Lennon, Saglam. And then I think we can now go to the general conclusion of the talk. So I think that we see that the anterior vermis and nodulus uvula are two distinct types of vestibulo-cerebellum, one which appears to be more spinal and the other vestibular. However, we clearly see evidence of internal model computation, which are distributed a little bit everywhere. So of course, we simply don't know how the entire computations are encoded. We don't know what every cell is doing, but we see a lot of things that make sense in the context of internal model. Some open questions. So we see that the nodulus and uvula seems to encode to distinguish TIL from translation, and then we seem to see translation selective cell in the rostral fascicle nucleus. So is it that this net translation signal are transmitted through the vestibular nuclei? That's a question mark. And likewise, are these external motion specific signal come in the vestibular nuclei coming from the anterior vermis? These are all open questions. I think there's a really interesting question, which is what is the point of convergence between these two vermis? Is that the medial vestibular nuclei? Well, nuclei, then that's a nice candidate. An interesting thing which has showed up recently, apparently there isn't any reference copy in the nodulus and uvula, and here I just have to smile because for so many years as a postdoc, I presented my work with Dora at SFN, and people were like, oh yeah, it's all nice, but everything you do in the nodulus and uvula, you know, you all do that with passive motion and you don't consider reference copies. And so to hear now that there might not be any reference copy in the nodulus and uvula, well, that's kind of funny. Welcome. And then, what else? Okay. Yeah. And then I think another thing that I hear quite a lot is when I explain the standard model of vestibular sensation, I get questions like why? Like, why do we have an internal model? And why does the brain discriminate TIL from translation? And why, and why, and why? And well, I think it's completely okay to ask this kind of question. And the best way to answer it actually is to go one step above and ask, why do we have a vestibular system to begin with? And the best way to answer this question is consider what happens when people lose their vestibular system entirely. And what you will find is that people without a vestibular system can live very well in everyday life, but when they go and walk on wet leaves after the rain, then this is when they fall. And so that's the idea that a vestibular system loss leads to postural instability on slippery surface, wet leaf, in brief, in any condition where you have a lot of motor error and where the environment is unpredictable. And this perfectly matches the notion that the vestibular system is a feedback mechanism inside of an internal model for motor control. I would again repeat that the internal model is excellent at explaining the impact of vestibular lesion, and therefore it practically probably has a lot of application to clinical cases and neuroprosthetics. And with that, I will conclude by saying that in my lab, one of my inspirations for doing marmoset has basically been to move towards motor control, and for sure marmoset are a great model for balance control. I'm also developing a marmoset postural platform analysis. Yeah, here it goes. Is the marmoset coming? Oh, this movie is not working. All right. And with that, I think I'm going to conclude, and I'm ready for some questions.
Thank you so much, Jean. Yeah, if you have questions, please go ahead and raise your hand, and I will call on you. I can start. You mentioned that the olivary input are different to the anterior vermis and the nodulus. They come from different parts of the olive. And you mentioned that from the nodulus recording that you showed, you demonstrated that the complex spikes appear to be modulated by acceleration. Um, what do we know about the input, the climbing fiber input, and how does it differ in terms of what its encoding is in the anterior vermis?
So I have not reviewed that extensively, but certainly an important question. So I don't know it myself exactly. So I know that the climbing spino-olivary pathways basically encode information from the spinal cord. So mostly, I assume, motor or proprioceptive information. I don't know the exact detail. Uh, I don't know that the Cullen group ever published anything about complex spikes. I don't know any direct recording in the inferior olive. I think there might be some complex spike studies in the context of locomotion. So, however, I have to say I don't know this by heart. I think that's really an excellent question, and really what the system needs is more studies about this entire circuit.
All right. Some questions from my lab. Go ahead.
Hi, really great presentation. Thank you. I was curious what you think about the beta phalangeal nucleus and what it might be doing. You put labels and there's studies on all the other regions, but that box seemed to be a little bit of a question mark.
Yeah, so it's really empty because I really had to go back to like peroxidase studies for the 80s in order to learn anything about it. So what can I say? Basically, tracing study will trace using anterograde or retrograde tracer are basically telling us that the nodulus uvula project to that little medial portion of the fastigial nucleus and not to the rostral fastigial nucleus. So this is important to know. Tracing study tell us then that in turn the beta nucleus projects to the medial vestibular nuclei, and that is pretty much everything that is known about it. One of the problems also with all of the brain recording studies that have been done, it's been done in monkeys, and in monkeys, you never really get a super accurate anatomy because you're not going to sacrifice your monkey every time you record the neuron, for sure. Once again, what we need in here, I think, are studies in rodents or in rabbits that combine really good, let's say, the appropriate protocol with modern anatomical technique.
Wonderful. Are there any other questions for Jean? We had a comment in the chat that Robin mentioned that she's in Kathy's lab, that they're working on climbing fiber recordings to understand the input to the anterior vermis. Wonderful.
Wonderful. Yeah, I look forward to seeing the results.
Excellent. All right. Any other questions? I think you gave such a lovely didactic and teaching style of presentation, Jean. I'm very grateful. Thank you so much.
Thank you.